good first. I think it's a really good, let me get this out of the window. There we go. Really good way to open the Tract-Anat-Club. I'll be talking about a history of tractography. And I have to thank a few people. First, Francois and Chiara for like what Francois just described, setting this up, setting up a really exciting series of speakers. And I'm excited to see what some of these future talks look like. And then I have to thank Carlo Pierpali, Derek Jones, Peter Besser, and Denis Lebihan for really, really excellent discussions, chats, email conversations over not just the last few weeks, but the last few months and years that are just fascinating to listen to, but also really provide the background and the basis of this talk. And if you haven't read it, I highly recommend reading the last 4 or 5 chapters of the Diffusion MRI. Well, actually recommend reading all the chapters of the book, but the last 4 or 5 are from pioneers in the field, perspectives from pioneers, and are just really fun to read. You get to see some handwritten notes, some correspondences, and really get into their mind in the late 80s and early 90s. So it's truly a fun read. So the whole theme of this talk is behind every one of these beautiful tractography images or these tractograms that we work with every day is really at this point 30 or more years of history that sometimes maybe we don't appreciate, sometimes we don't know about. And it's always good to know where we came from, to know how we got to where we are today and how we overcame some challenges over the last 30 years. So I'll give a very brief historical overview of tractography. I actually plan on talking for less than half an hour. So please interrupt me at any point. Please raise your hand if you want me to zoom in on papers or figures or abstracts. And then we have plenty of time to chat about this after as well. I'll give a little bit of how some of the motivation and inspiration from some of these early researchers, how some of the early tractography studies, what their applications were, and just as interesting, some of the challenges they faced that are still very much challenges we face today. And I want to point out that I'm kind of going into this assuming you know what tractography is and kind of how it's applied. I won't really describe the tractography process or too much nomenclature terminology as I go through this talk. So what you're going to see are a few different timelines. We're going to go through the timeline of visualizing and orientation and anisotropy in the central nervous system, the timeline of visually, qualitatively assessing white matter structures using diffusion. Then we'll get to streamlines and what we think of as present-day tractography, characterizing uncertainty or how confident we are in these structural connections, dealing with some problems and challenges in the field of tractography. And then I'll kind of cover at a very high level some of the history of some applications of tractography. And you can see this is kind of chronological. There's some sanity to the ordering here, but I will kind of be jumping back in history as we go to each timeline. And just as exciting, I have a couple branches or offshoots. So the very first offshoot actually happens completely off this timeline. In 1985, this was going through some of the ISMRM historical archives. I just thought this was really interesting to see the very first diffusion magnetic resonance images. These were actually presented in 1985 at London at the 3rd annual ISMRM, or SMRM, Society of Magnetic Resonance in Medicine at the time. We have work from Denis Lebihan actually showing diffusion contrast in an in vivo human, showing our PGSE sequence, introducing the B-value and apparent diffusion coefficient. And actually, it's not the first introduction of this, it's actually reintroducing it to the field of MRI from NMR. We have work from David Taylor and Michael Bushell from Guildford, Connecticut, doing diffusion MRI on chicken eggs and actually calculating the diffusion coefficient in egg yolk and egg white. And we have work from Klaus Merbold in Germany, actually showing using stimulated echo sequences, and in the abstract showing diffusion, contrast, and phantoms, and then presenting on plants, animals, and human extremities. And well, of course, this isn't explicitly related to tractography. There's, of course, a huge amount of work that I can't cover, work in readout and figuring out diffusion contrast, diffusion weighting, eddy currents, motion, all sorts of things. But what I really consider the birth of tractography is this 1990 study from Michael Mosley and colleagues from UCSF. Around this time, they were studying diffusion-weighted signal in cerebral ischemia, and they took the time to actually measure the diffusion coefficient along a variety of axes or in different directions. And they had this fascinating discovery that diffusion of the central nervous system was anisotropic. It was not the same in every direction. And immediately they recognized how exciting this was in the potential, where they now see that it's possible to determine the orientation of white matter tracks from the knowledge of the directional dependence of diffusion. And this sentence alone kind of encapsulates the entire field of tractography. This is what all of tractography is based on today. And almost immediately, this new exciting contrast in white matter was taken advantage of. Researchers said, oh, how can we visualize this and appreciate orientation and anisotropy? And the very first diffusion encoded color map was introduced by Philippe Duec and Denis Lebihan, where they said, well, if diffusivity varies according to fiber direction, then we can infer the direction of fibers by measuring ADC in multiple directions, and we'll say that the fiber direction is the one with the highest ADC. And that's exactly what they did. They just colored their images based on the direction of highest diffusivity. And I found it really interesting. In this paper, they actually said diffusion anisotropy is best described mathematically as a tensor. They even described what the diagonal and off-diagonal terms of this tensor meant. But at this point, we didn't really know how to measure the tensor. So what they're showing is actually more a vector of diffusion. They really just measured diffusion in two directions, for example, left, right, and up, down, and then colored the image based on which kind of the ratio of these or which one had the greatest diffusion. So it turns out that Peter Basser was looking at this poster from Philippe and Denis at a science fair or a science festival at the NIH. And thought to himself, you know what, diffusion really is best described as a tensor. We need to figure out how to measure this. The back of that diffusion MRI book actually has Peter's both email and handwritten notes to Denis. That's really interesting to read as he finds out that we didn't know how to measure a tensor at this point. And then you can go through the process of how they figured this out and introduce this in 1992 at ISMREM. Where now for the first time we have a way to model or encapsulate this diffusion process in a quantitative way. So like I said, they presented this at ISMREM. They actually did this first in a pork loin sample, where they measured the tensor in the sample of pork loin. They rotated the pork and showed that the tensor rotated appropriately. And here, Peter even describes the process of tractography. One can envision 3D fiber maps or diffusion ellipsoids displayed in each voxel, connected like link sausages that follow fiber tracks. And that's really what we do, again, with tractography. These sausages are the vectors from our diffusion tensors. And all we need to do, very simple, right, is find a way to connect these sausages to get from point A to point B. And we're going to talk about some of the innovation that had to happen to connect these sausage links, maybe in the next 5 or 10 years as I go through this talk. So back to color maps. A lot of ways to appreciate orientation and anisotropy were proposed in 96, 97, 98, particularly at ISMREM. We have work from Aziz Ulig, actually coloring the image based on in-plane and out-of-plane orientation. Work from Derek Jones that colored the image by in-plane orientation and its brightness by out-of-plane orientation. And then, oh no, we have more color methods from Carlo Pierpali that ultimately transformed into this work with Sinisa Pajovic. That's the very famous red, green, blue diffusion encoded color map that we still use today, where red, green, and blue represent tensors oriented right, left, superior, inferior, and anterior, posterior. And it's pretty obvious right away just by looking at this why this became so popular and so easy to look at. You can appreciate directionality, you can appreciate anisotropy, you can appreciate kind of the borders of some of these white matter pathways. And even in 1999, you can see the beautiful, small, intricate pathways of the brainstem. And immediately it was recognized how important this was. Even without tractography, we can see differences in pathways. For example, degeneration, where it's pretty clear, hey, there's less blue or less bright blue in these cerebral peduncles. One really interesting story from this is that when they introduced this color map, they were comparing to existing color maps, basically conveying, oh, ours is a little more intuitive, ours is a little more visually appealing. And they implemented a few other ones. They happened to implement that Derek Jones one that we saw on the last slide, but completely incorrectly implemented this so that the colors were complete nonsense and didn't mean anything. It turns out they issued an erratum apologizing, saying, oh yeah, the Derek Jones one does implement antipodal symmetry the right way and should make sense. But they ended up also implementing this erratum wrong. Which didn't matter because they ended up working together at the NIH a couple years later and published a great series of studies over the next 5 or 10 years together, many of which we'll see. So even though we're not at tractography yet, even in these very early years, this is a 1996 study from Carlo Pierpali, it was recognized some of the challenges that we're going to face when we get to tractography. Here, they pointed out the presence of fibers that run, oh, sorry, you can't see my mouse. The presence of fibers that run in multiple directions within the voxel, and that this pattern may be presented in a majority of voxels in white matter of the brain. And this problem of crossing fibers, even though, again, we're still not at tractography, is really going to pop up right around the year 2000 to 2005. Is going to be a major theme of the history of tractography in the rest of this talk. So, at this point, we can visualize voxel-wise anisotropy and orientation. It was also immediately recognized that you can appreciate entire white matter structures just by being smart about which direction you sensitize your sequence to diffusion in. This is a very early study from Mark Duran and group from Hammersmith Hospital in London. Even in 1990, the same year that the Moseley studies were published, they showed that diffusion-weighted images alone enable the ability to visualize normal and abnormal white matter tracks, enables identification and characterization of entire white matter pathways, and amazingly applied this to about a dozen diseases and pathologies, hematoma, glioma, epilepsy, agenesis, and showed that without any math, without any fancy tractography, this contrast is useful for identifying normal and abnormal white matter. Very shortly, words like neurography showed up, where a neurographic image is analogous to an angiogram, but highlights nerves rather than blood vessels. This is one of the first examples I was able to find. It happens to be in a rat tibia, where the tibial nerve is very nicely highlighted because of diffusion restrictions within the nerve. And in the very next year, the very first patent was issued for segmenting white matter structures with diffusion MRI. This was issued to Aaron Filler from Seattle University, the same group that did this neurography paper. It's really weird. The patent says 1996, but I found that it was issued in 93. And you can already appreciate, even though it's, again, not tractography of today, they're just saying that if you sensitize the sequence to diffusion in the right direction, you can nicely highlight these entire white matter pathways, in this case, the cortical spinal tract. And in the same year, 1993, the word tractography very first showed up in the literature from a Japanese group, Yasutomi Kinosada. And very interesting, not just the word tractography, but also tractogram. Everything but the abstract is written in Japanese, and I've really struggled to translate this and would be super enthusiastic if someone was able to do this. But luckily, we can still read the abstract. And it's really interesting that they say tractography has two steps. One is to obtain diffusion images sensitized along axes appropriate for detecting the intended nerve fibers. And the other is to extract the structure of the nerve system from a series of diffusion-weighted images by maximum intensity projection method. So you can see that what they're doing is picking a pathway of interest, sensitizing to diffusion in the appropriate direction, and then acquiring many slices and doing a maximum intensity projection. And it's clear you can appreciate these pathways or this unique contrast in white matter at a very high SNR that could be useful for, or ultimately we know is useful for studying the central nervous system. So at this point, we can visualize local orientation in anisotropy. We can visualize entire white matter pathways. Now, we really want a way to form connections from point A to point B, and that's where streamlines come in. But it ended up being a really challenging problem to really step along these vector fields that diffusion tensor imaging made possible. And talking to a few of the pioneers, they pointed me to some literature that was around at the time, or even around 30 years prior, 30 or more years prior. For example, in meteorology, they actually have a more challenging problem where they don't just have discrete points, they have a really sparse set of discrete points and need to figure out some variable or some vector field within this set of very sparse points. Also, luckily, the field of computer vision was also proposing ways to visualize second-order tensor fields with hyperstreamlines. So our field learned a lot about how to do this and how to interpolate between points, how to connect points, and introduced the very first streamlines. The very first streamlines were actually introduced in 1995 by Van Wedeen from Massachusetts General Hospital. It's actually an OHBM abstract, where amazingly he actually did commissural and projection streamlines. We have work a couple years later, 98, 99, 2000, work from Susumu Mori doing this on an ex vivo mouse model, work from Peter Basser showing how to do this in a simulated vector field, and work from Derek Jones that's not actually creating streamlines, but connecting voxels based on orientation and similarity of orientation to create these maps of structural connectivity in the brain. Using logic that's not quite how we generate streamlines, but using logic that's very similar. And again, what separates these studies is how they choose to step along the vector field or propagate these streamlines, where most of them just say something like the direction of the path at the next time step is determined by the vector field at or around the current time point. And how they kind of interpolated these vector fields or said, maybe we should step to the next point or not, is really what separates a few of these studies. A couple years later, 99, 2000 is where the first peer-reviewed papers came out. We have work from Cyril Poupon doing fiber tracking by minimizing some bending energy and linking voxels that are connected. And they ended up liking this to a plate full of spaghetti noodles. And very interesting, they described another challenge that we're going to face today is the not just the crossing fiber problem, but a bottleneck of fiber pathways, or the bottleneck problem, where multiple pathways converge into the same area in the white matter and then diverge. And they recognize that this is going to be a very challenging problem to solve for the field of tractography. They also, very interestingly, already were segmenting U fibers or superficial white matter. Another structure that we know is challenging today because of partial volume effects, and that we're still iterating on and perfecting today. We have work from Thomas Conturo. That's one of the first examples of generating a whole brain set of streamlines and then doing the virtual dissection procedure using manually identified or expertly identified regions of interest to identify subsets of tracks that link these regions, showing the logic to do this for all sorts of projection, commissural, and association pathways. We have the method from Susumu Mori, introducing the FACT method, the fiber assignment by continuous tracking, that became widely popular over the next few years because it was so simple and so intuitive to implement. And then we have work from Peter Basser generating images that are just as beautiful as the ones we generate today, but also pointing out more challenges, what they called singularities, for example, where fiber tracks cross or kiss or branch or merge. And I really find it remarkable the insight that they had as they were generating and developing these algorithms, already the insight of where they fail or what's needed next in the field. So now that we have these streamlines, we want to use them in some way. We want to quantify them. We need to know how certain we are that they represent something meaningful. And the field realized we need to be able to characterize uncertainty in this process. And they generally characterize uncertainty in two processes: uncertainty in fiber orientation, in estimating the eigenvector from the tensor, and uncertainty in that streamline propagation itself. So even in 1997, how important it was to estimate uncertainty was recognized. We can calculate a tensor, we can get an eigenvector. And Peter Basser first showed this idea of a cone of uncertainty, where even though we calculate it, maybe just noise in the image, we're only really 95% sure that it's somewhere within this cone of angles. They went on with Sinisa to actually, so at this point, they're using Gaussian noise in Monte Carlo simulations. They did a bootstrapping approach to show uncertainty in all the tensor parameters a couple years later. And then Derek Jones very nicely showed how to do this using real empirically acquired data that takes into account not just image noise, but physiological noise and issues, but really nicely shows the spatial variability of uncertainty, how it relates to anisotropy, and even said and really advocated for using uncertainties in fiber orientation obtained by this method to be incorporated into tractography algorithms to make probabilistic track maps. And that's exactly what happened. The very first probabilistic or probabilistic-like algorithms were introduced in 2001, 2002, 2003. We have work from Martin Koch from Max Planck Institute, where they actually implemented a particle jumping strategy, kind of similar to the diffusion process, where they asked how likely it is for this particle to jump to a given voxel if it starts at some other voxel. We have work from Timothy Behrens using Bayesian uncertainty to estimate probability distributions, and actually using these probabilistic maps to segment the thalamus into anatomically meaningful regions. And then we have work from Jeffrey Parker and team relating uncertainty to anisotropy to segment specific white matter bundles and give us a measure of how confident we are that bundle sits in this voxel. And already they were advocating for caution when interpreting this index. This index should be interpreted as an index of how confident we are, given the assumptions we made about the nature of the diffusion data, that a route of connection exists between the start point and any given point within the brain. So we have these algorithms that generated the streamline simply by stepping along that vector field. We have these algorithms that are kind of doing this, maybe a bootstrapping or a probabilistic type approach. And the first time that these algorithms were, even though the word probabilistic had shown up before, the first time there is a clear distinction and terminology applied to this was actually this 2004 abstract from Derek Jones. Is the very first time I could actually find someone describe deterministic, describe probabilistic, describe their differences and similarities and how they may be used together. So I thought this was a good point for the second branch or aside. I found it really fun to read through these historical archives. And in 1997, there's actually 6 consecutive abstracts that are all just fascinating reads and important to our field that show up in the just quite literally consecutively. We have Ono, Carlo's other method. We have the Derek Jones color mapping method. Peter Basser showed the analytical calculation for the diffusion tensor given 6 directions. And it was work like this that I believe was truly important for the field, for people like me that aren't experts in math or linear algebra. that shows me a way to actually go ahead and program or analytically calculate this thing in a very simple, very intuitive way. We have the cone of uncertainty from Peter Basser. We have how noise propagates into biases and eigenvalues. And we even have how important it is to rotate your b-vectors when correcting for motion in diffusion MRI. And this work from Wayne Daniels. So now we have streamlines. We kind of know how confident we are or can characterize uncertainty. Now we need to solve some challenges or problems in tractography. Like I said, very early on, it was recognized that this crossing fiber problem can be an issue for diffusion tensor imaging, and that it maybe occurs quite often. It wasn't actually until 2013 that we quantified the prevalence of this issue. This is a beautiful study from Ben Jurison, where they quite convincingly showed that this challenge or this problem occurs in 75 to 90% of white matter voxels in the brain. So we definitely need to deal with this problem. And there were many methods proposed to deal with this problem. One of the very first is this work from David Tuch, where he presented high angular resolution diffusion imaging, or HARDI. And what he said was, well, we need to acquire more than 6 directions. Let's acquire many directions and just show the diffusivity profile in every direction. And hey, look, it's quite clearly not Gaussian, so we need to develop methods to deal with this. And many methods were developed. We have work, diffusion spectrum imaging, work using the diffusion profile. We have the original spherical deconvolution from Adam Anderson, where for the first time now we're actually estimating the fiber orientation distribution instead of the diffusion orientation distribution. So this is actually the very first glyph of a fiber orientation distribution, presented in 2002 ISM Rem. This was published a few years later, and I actually bring up this paper not for the spherical deconvolution part, but because I find the appendix quite a great read. It's an excellent review of anything related to spherical harmonics. It showed how to relate the diffusion signal to the fiber orientation distribution. It showed how to perform deconvolution with spherical harmonics. It actually went further and showed how to do cue ball imaging, how to integrate functions over a sphere. And in the appendix is a method for calculating correlation coefficients for spherical functions, which is going to become important when we compare some of these methods or maybe validate it against some sort of ground truth. We have more methods. We have persistent angular structure. We have cue ball imaging from David Tuch. We have a different method and actually named spherical deconvolution from Donald Tournier. We have regularized cue ball imaging that's the much more common version that's of the original that's still very much implemented today. And then we have super resolved spherical deconvolution, again from Donald, that I'd argue is likely the most commonly used reconstruction method for tractography today. So we have a ton of methods. The good news is that we're quite good. All of these methods do quite well at dealing with crossing fibers. I really like this study, 2014 study from Alessandro Deducci. It was really a big community effort where they created these ground truth simulated data of all sorts of crossing fibers, and then implemented a couple dozen methods. And you can appreciate that all of these methods quite well reflect the directionality within the ground truth. All of them, of course, except for DTI. So the good news is, even though there's many methods, they all do quite well and will all likely do quite well for tractography processes. So one more aside, now that we're in 2003, I wanted to bring up David Tuch's thesis. I'm always impressed with the amount of content in this thesis. He introduced multi-tensor imaging, fitting 2 tensors to deal with the crossing fiber problem. Even though it was recognized that diffusion within the thalamus was different in different nuclei, he presented the first quantitative thalamic parcellation using clustering of diffusion properties. He presented diffusion spectrum imaging that wasn't published for another 4 or 5 years. And he introduced cortical-cortical connectivity mapping. Even though whole brain streamlines had been described and words like connectogram had been used quite often, this is the very first matrix describing some measure of connectivity from region to region. And in the same work, he did U fibers and showed a way to pull out the centroidal bundles. And then, of course, he introduced cue ball imaging that, like I said, is still used today. And I really like the way that this thesis ended. It's not actually tractography specific, but he asked a few really intriguing questions. Where does diffusion anisotropy come from? What's the trade-off between anatomical and diffusion space resolution? What are the roles of diffusion time and gradient duration? What does contrast and noise mean in diffusion projective space? And what's the relationship between the observed diffusion and underlying tissue structure? And these are questions that don't just form 6 different theses. It forms the basis of what we're all working on today and what the entire field of diffusion is trying to answer. So we're finally on applications. We can make streamlines. Awesome. What can we do with them? Well, I generally kind of separate 2 different applications: tractometry, which I consider any quantitative analysis with streamlines, and then connectomics, which is studying the brain as a network. And the very first example of quantifying some feature of streamlines or bundles of streamlines is this 2003 ISMRM abstract from Derek Jones and colleagues, where they studied schizophrenia and schizophrenia-like psychosis. And they said, well, we have these methods to virtually segment, for example, the superior longitudinal fasciculus, the cingulum bundle. Let's actually just ask what some features, the anisotropy or diffusivities, are within these very well localized areas. And they showed, comparing controls to patients, that there was, in this case, some decrease in anisotropy in biologically significant regions. And this type of analysis was almost immediately applied to all sorts of diseases and disorders over the next few years, and is still applied today. Maybe more interesting, you can actually quantify features along streamlines. And one of the first examples of this was actually quantifying uncertainty along streamlines for the purposes of probabilistic tractography. You can also quantify something like anisotropy along streamlines. And even though tractometry had been done, the word first showed up in 2011 in this abstract from Sonia Bellis, where they showed that you can not only pull out features along streamlines from diffusion contrast, you can do this for any contrast: myelin water fraction, QMT, relaxometry, non-MR contrast, and compare across bundles and compare across features. And I really think tractometry became popular with the introduction of automated bundle segmentation and analysis methods. One of my favorite examples of this is AFQ from Jason Eatman, a long track fiber quantification, where it's not only beautiful pictures, it's an automated way to segment the structure, to create this long track analysis and then compare controls and cohorts. Many great examples of automated segmentation exist using anatomical priors, using similarity or reproducibility across populations, comparing shapes to expertly delineated bundles, using expertly placed manual regions of interest in standard spaces, and even using deep learning to segment these structures. And I think all of these methods were crucial for end users to ultimately use tractography in a reproducible way. So onto connectomics, we saw that the very first cortical connectome was introduced or shown by David Tuch in this ISMRM abstract and later in the thesis. But this field really exploded with this Patrick Hagman study from Lausanne, Switzerland, where he showed not only how to do tractography and create the streamlines, but how to integrate it with our structural information, creating this whole brain structural network, showing the matrix that we typically view today, and graph theory measures that provide us insight into the brain as a network. And this really propelled our field to try to make anatomically meaningful streamlines. And this is just a small subset, but there's a huge number of methods that try to make streamlines somehow related to some measure of structural connectivity. Methods doing global tractography informed by microstructure, filtering streamlines based on the signal, based on anatomical priors, based on, again, microstructure informed tractography, or even based on assumptions about how other contrast should act along or across streamlines. All of which are very often used today and still very active area of research for studying the brain as a network, as well as actually studying individual bundles. So that's all I've got. I walked through 6 ideas: visualizing orientation, appreciating white matter structures, creating these streamlines, characterizing their uncertainty, overcoming challenges, and then applications. And I'm excited to see what 2024 and the future brings, and hope we can have 15 or 20 minutes of really great discussion.
Thank you very much, Kurt. That was an amazing extended version of your talk from the retreat. I really liked it. The floor is open for question. I could start by just asking, how stressed are you that you missed something and somebody in the comment will just correct you on it? Are you stressed about that?
Very much. I was watching your faces, and when I'd say something, I hate saying the word first, but the first this or the first that. But I'd watch your faces when I said that, and I was trying to look to see if someone reacted and then see who reacted and why they might have reacted. But I didn't see any bad reactions, just saw some smiles, which is great.
Well, I think that you added quite a lot of content that I didn't know about, so I cannot fact check you. I trusted at it, but if there is anyone that knows something that no one else knows, a secret abstract from an obscure conference, it would be very nice to get feedback. We have 80 people connected, so like maybe one of them can add a tiny bit of information. And if there is any question, I guess you can unmute yourself, ask the question. You can write it in the chat. Is there anyone?
See, Alex.
Oh. So amazing talk, as usual, Kurt. A quick first abstract that might be also cool to put in, and again, it's Dave Tuch, of all people, right? Is also about probabilistic tractography. He has an abstract in 2000 at the ISMRM discussing how fiber tractography methods currently in those days, you know, the deterministic ones, were not amenable to probabilistic interpretations of the tract solutions. And then he posed the question, what is the probability associated with a particular tract solution? And I just quoted him. Okay.
2000?
Yeah, ISMRM 2000.
So
that's yet another, you know, credit to him in a way.
That's a good one. And the entire 90s is really easy to go through. They seem to group the diffusion abstracts together. And you can go one after the other and you find something fascinating after every single one. But I don't think I did 2000. I think I was stuck in the 90s because of all the stuff in 97, 98, 99.
So when you split, quick question, when you split in your 6, was there any extra category you wanted to add, but you cut out because you were limited on time? Like, I'm curious because like, it seems like you did so much research for that. Like it's, I don't know how much stuff you went through, but it's very impressive. But I'm curious if you cut anything that you're like, that's too much.
I really wanted to do atlases. Because the history of so the structural atlases existed in right, like many of us use the JHU white matter atlas, and not just atlases, but atlases and who created these protocols. So in the early papers, right, the Conturo papers or things like that, there were protocols, but I want to know what they thought of when they were doing this. Or we even had the book, the human brain connections. Like, what was the logic for when you said, well, of course we know where the SLF is, but do I want a region here and here, or here, 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 here? And I don't know who to answer those. It may not have been the first author, and it may not have been an anatomist even on those papers. It was just guess and check. I don't know.
That would be a very interesting subject for next year. Trying to find someone to do like a similar like 6 or 5 categories of like brain anatomy, and like, you know, the first is maybe like dissection, the other is tracing, the other like having kind of a big 200-year review of like anatomy. It's probably harder than tractography. It's a bit longer timeline, but it would be interesting to like simplify it for non-neuroanatomists in a way that we can understand where it's coming from. I see that Max raised his hand.
Yeah, well, maybe to follow up on this topic, thanks, Kurt, for the great talk, but where would you place the validation component of tractography on this timeline? And lots of us have worked on tractography challenges, but a big topic in this club will be neuroanatomy and maybe neuroanatomy validation. And I know you've worked a lot on it during your thesis. So if you had to put a validation timeline in there, where would you start? And should we have a history of tractography validation at some point?
Yeah. So I see a few faces on this call that are much more appropriate to answer that question than me. But I can tell you that most of what I typically reference for tractography and fiber orientation validation is really kind of 2010 and later. But I'm sure it existed before that. That's a really good question. Or like, what were the early attempts other than saying, yeah, this matches what we think it is in the brain? I can think of a few studies that compared it to dissection, or actually there's a bunch of really great simulation studies already in the late 90s, or 2000, 2001, showing the uncertainty or looping around or in simulated fields. So I guess that's validation, or at least understanding limitations of the process. But I do see some faces that can better answer that question.
Yeah. Any comment from the room? Any senior people in the room? More experienced people in the room?
Well, I think that that we did this pig brain that was in President ICMM in 2005 or something like that. And I know there's a study doing manganese in the optic nerve. I can't remember who was it a few years before, but I think this was the real brain connection validation studies. There's one in monkey also at the same time.
Cool.
Out of curiosity, like Maxime, in the book of tractography that will come out, is there a history section?
I'll let Flavio comment. There's a bit of history spread around the book, but Flavio, do you want to answer this one?
Yeah, I wasn't able to find the microphone, sorry. Yes, there is, but is with outside tractography. So is how we study connectivity before the advent of MRI. We talk about a little bit of PET and fMRI, but we haven't covered this part. We have done all the postmortem dissection or axonal tracing, electrophysiology, but we stopped there. No, we haven't done that.
I'm saying, Kurt, not that you have not enough paper, but like that would be very, very interesting to put on, like, you know, in a formal manner with a timeline, maybe pull some, like, you know, like Alexander just told us about an extra abstract. That could be very interesting to put in paper. And I think having a validation or neuroanatomy history, maybe from what is in the handbook of tractography inspired, that could be nice for next fall, because I'm very curious in that too. Like, it's a simple talk in terms of technical knowledge, but in terms of content, it's very, very interesting.
Yeah, I think it's interesting to see in one place as well. But a lot of this really is from the last 4 or 5 chapters of the diffusion book, but it is focused, that's more diffusion focused. Whereas I don't think we've compiled, right, those 98, 99, 2000, just the very first streamlines anywhere. And those are just fun to look at. What were the first streamlines? So I think it would be cool to see.
I wonder if someone will find earlier Streamline. Is there any other comment, question, observation, suggestion from people in the room?
Well, I would like to ask with Kurt, I think it's amazing what you did here. But what's interesting is that you include also all the abstracts. And when we do the publications, it's not always that you get all these very, very early ideas into these publications. So this is what the cool thing about this storyline is really, what is it where you start these ideas of what that will come out with papers later on, or maybe not coming out with papers, and then something that we still are working on.
Yeah, that was fun to see too. You saw the unfinished ideas, or you saw the cone of uncertainty before the beautiful spatial visualization, or the importance of correcting for head motion before the there was so much fun history in here. The Daniel Alexander it's critical to correct B vectors paper that we all cite is actually the back of this paper. If you print the Mackay abstract, it's actually the one after it. So when you print it, you have to print the title page of it. But 2 years before this, it was shown in ISMRM that I'd never seen before. So the abstracts really are just as much or more fun than the papers.
Any
A lot of
other...
plants.
Oh, sorry.
Well, I'm taking notes because this is interesting. This to me are ideas on how to communicate better, educate ourselves better, and maybe that's food for thought as a group for us as a community, and as we start growing the community to think beyond the PDFs of papers and see how we can share these ideas better.
Just Kurt, it was a great job, thank you. Just people might be interested that David Tuch is still around. He's not in MRI, but here's his LinkedIn. And maybe it might be fun to get him along if he's interested, and you could interview him and find out what he's up to now and what he didn't manage to fit into his 7 chapters of his thesis, because I'm sure there was extra ideas. So anyway, he's still alive and kicking.
That's a great idea. I think maybe we could get his draft for his unfinished chapter and and finish this. The secret chapter that no one knew.
And actually, I'll try and find it. I don't know if you still have it, Kurt, but the recording of the NIH conference in 2000, you can actually see Contoro presenting the ideas and Maury, and I think, I can't remember if Dave Tuch was there, but Van Wedeen was there. And if I can find the link whilst we're talking, I'll put it in the, before we get kicked out of the Zoom, I'll put the link.
I will also put it in the chat. I have it. It's 7 hours of great lectures, and I unfortunately did not make it through all of them, but I'm about 4.5 hours in. I think this is it.
Actually, that's a good point. I don't know if we are going to get kicked off in 5 minutes. So just in case, I'm going to say that this was recorded. I'm going to discuss with Chiara if we put the link for download in a spreadsheet so people can have it. Do we put it on the website? Do we put it on YouTube, on the channel for the International Society of Tractography? We're still figuring this out, but next week at least should be in the spreadsheet, at least, so everybody can see it and share it around. And we're gonna have a more permanent solution for later. Also, I mentioned at the beginning that next month on the 20th of June should be Silvio Saribo giving a talk about more clinically oriented for neurosurgery, stimulation, tractography, and anatomy. We should send an invite as well as description and bio to the email that are on the spreadsheet. You can share that around. We were 80 on the call, so I think we are fine with our 100 limit, at least for next month. We're gonna update that maybe for next fall. And before we get kicked off, I just wanted to thanks Kurt for this amazing talk, and also like opening the first Tract-Anat-Club, which you should include in your timeline as a a flag in 2024 at the very end. So thank you very much for accepting to break the ice. We can continue talking. I don't know if we're gonna get kicked off. I just wanted to be sure, and I'm gonna stop the recording so you can say nonsense if you want.